Hello, welcome to the next A-level lesson from the conscientious biologist, Ben Gallagher. This one's the third one in the nervous system series of lessons. This one follows very closely on from nervous system 2, which was about the action potential, which covers how that nerve impulse is generated. This one's saltatory conduction, which links to how the action potential was actually stimulated in the first place. What was the initial creator of that? Now we're going to look at that in very basic theory to give you a really good understanding of what saltatory conduction is. Then we're going to put the complex mechanism behind that. I've also tagged onto this one some information on threshold potential, which is essential for understanding saltatory conduction. And then because we do a full overview of the whole nerve impulse, filling in the gaps from the previous video to complete the graph, I'm adding a little bit on the refractory period as well. So let's get started on that. As always, if you haven't subscribed to my YouTube channel, please do so. And also you can find my stuff on Instagram and Facebook. Thank you. So we're going to go quite quickly through these first two slides because it's just to give you the clear understanding. So this is the basic principle behind saltatory conduction. So if I put an unmyelinated neuron there, that's a neuron without any myelin, so no Schwann cells wrapped around it. This one's a motor neuron, look. If we're going to send a nerve impulse along there, you can see it goes from point to point to point to point to point to point until we get to the end. It takes forever, okay? But each action potential is stimulated by the positive action potential that went before it. So if this bit went positive, this bit will go positive. If that bit goes positive, that will go positive. It's just like the Mexican wave that we talked about in the previous example. If the person next to you goes up, then you go up in response to them. Now with saltatory conduction, you can speed that up massively by having the Schwann cells that wrap around your neuron, making that myelin sheath and just leaving short gaps in between them that are called the nodes of Ranvier. Now in the nodes of Ranvier, you've got the exposed membrane through which your ions can move. So all we're really talking about with Schwann cells is they prevent ions from moving in or out of the neuron. And to get an action potential, you have to be able to, if you remember from the last video where we did this sort of funny little dance move thing, if sodium's going to come in to create your action potential, potassium's then got to go out, then they've got to switch. It's all about these ionic movements through membrane. Well, if the membrane of the neuron is wrapped up in layers of membrane, which all the Schwann cells are, then you can't move ions in and out. So you're limiting action potentials to the nodes of Ranvier. Now what that means is you get this situation where bang, 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 and we're at the end already. Because the impulses can only happen in the nodes, there's far fewer of them, they jump, so it goes much quicker. So the basic principle of saltatory conduction is it's a way of massively speeding up a nerve impulse. And of course, faster communication around your body is going to hugely increase your survival chances. Let's go straight on to the second slide, just to kind of hammer this point home and make it a little bit more visual for you and link it to something that I've shown you before. So if I pull up, same neuron, but remember we talked previously about the signal needing to go from A to B, but it's not something traveling along like an electron and electrical wire, it's this wave of excitation. So you know that you've got this happening in an unmyelinated neuron. Each bit of membrane next to each one needs to have an action potential. Now, if I put that same thing up below, that took 6 seconds, by the way, to move along. If I put those same ones below, but I wrap some of that area in myelin sheath, so in the Schwann cell, so each one of these blocks is representing a Schwann cell, then you can see that we're only left with 4 of the little faces, which are representing ions. Technically, they're representing, well, it should be the other way up, but there'd be sodium ions sort of getting excited, causing the action potential and coming back. And now when we get a signal, it can jump up and down much quicker, and that only took 2 seconds. Now, I've tried to time those as accurately as possible because in reality, the speed in an unmyelinated neuron, so that's the middle one there, is about 30 meters per second. Now that's still extremely fast. But if you have saltatory conduction in a myelinated neuron because of the Schwann cell, so the signal jumps, then it increases to 90 meters per second. That's 3 times faster. So saltatory conduction is so important for speeding it up. And if those myelin sheaths break down, that's when you can get various types of neuronal disease, motor neuron disease, because the signal gets lost, it goes too slow, the communication pathways get broken down. Now, really important at this stage for me to tell you that there's actually 3 factors that have an effect on the speed of an impulse. Saltatory conduction is one. So having myelinated neurons is really, really important. Okay, if they're myelinated, you can have saltatory conduction. We've already said it goes about 3 times faster. The second factor, though, is the diameter of the axon. Thick axons allow for very fast nervous signaling. It's because it's easy, you get less leakage of the ions if there's volume inside with which those ions can be retained. So it's about leakage there. Don't worry about that though. All you need to worry about is wider, thicker axons carry signals faster than narrow, thin ones. And the third factor is the temperature. That should be an obvious one because temperature affects the speed at which all particles can move. And a lot of the action potential, a lot of a nerve impulse is reliant on facilitated diffusion. Think you've got facilitated diffusion of sodium ions coming in, then you've got facilitated diffusion of potassium going out. The active transport pumps then need to move it back round, but of course all particles go faster if it's warmer. So if you've got warm neurons, the signal will travel much faster than cold neurons. Our body temperature is 36.6 degrees. That's a good temperature for keeping relatively rapid neuronal communication. Take a screenshot of this one. It's got some really important information on it, but now let's get into the massive detail of saltatory conduction and how it links up to everything that we've done on action potentials. So, detailed mechanism. What I'm going to do is pull up an axon on screen. So it's just part of an axon of either sensory or motor neuron, it doesn't matter. And I've put Schwann cells covering it. I've made them fairly transparent though, so we can still see through to what's happening in the axon. Now, if you look on here, we could class the very end over there as maybe a node of Ranvier. Then we've got 2 very clear nodes of Ranvier. Maybe this is another one above me here. But these are where the gaps are between the Schwann cells, so this is where we're going to get impulses. Now, all of those locations, let's just mark the first 3. It's going to be at resting potential because you've got the sodium potassium pumps working, kicking out the sodium, bringing in the potassium at a ratio of 3:2, making the outside far more positive than the inside. So the inside relative to the outside is at -70. That's why we've got this -70 resting potential. Now, sodium should all be on the outside because of that pumping mechanism. So to create an action potential at the start here, we've got sodium coming in. That changes our -70 to +40. How that initiated, we'll do in the next video, but let's just link this to what we're doing now for saltatory conduction. That positive +40 there needs to send a signal to the next node of Ranvier. Now what happens is that some of that sodium that came in diffuses down to the next node of Ranvier. And you need enough sodium to move down to increase the potential in that node to -55 millivolts. -55 millivolts is known as the threshold potential. A threshold, if you've never come across that term before, a threshold is kind of like a barrier that must be crossed. It came from when, way back in the olden days, people used to put straw or rushes down on their floor instead of carpets, and they'd have a bit of wood in the doorway to keep the threshings, which is the straw or the reeds, inside the house. So the threshold held the thresh in, something you had to cross to go into somewhere. That's exactly what this is. It's a line, a voltage that has to be crossed to trigger the next thing. So enough sodium needs to come down to go from -70 to -55, because at -55, the voltage gated sodium channels can open in that node of Ranvier. This is why they're called voltage gated, because the gates open in response to a voltage, and the voltage they respond to is -55, the threshold potential. So as long as the sodium diffused from the previous node of Ranvier, then it's going to raise the voltage from -70 to -55, so resting potential to threshold potential, which will trigger the opening of the voltage gated sodium channel, so sodium can now flood in at that node of Ranvier to create an action potential there. So that's how we jump from way over that side to an action potential here. And now, of course, the same thing can happen. Some of that will diffuse down, raise the to the threshold potential in this node of Ranvier, which will trigger the influx of sodium here, which will give you a new action potential here, and then that will carry on and so on and so forth. Okay, so saltatory conduction is done by a localized circuit of sodium moving down, diffusing down through the axon, and then triggering threshold potential to create action potential, and that would just get passed on. Now, please remember, at each one of those subsequent nodes of Ranvier, as soon as that sodium moved in and some of it diffused down, they're going to straight away be starting to recover. So they'll be kicking potassium out, and then they'll be switching them back round again. So from the little kind of dance I showed you in the last video, where we had sodium came in, potassium came out, they switch. I want you to add to that now, sodium comes in, starts to diffuse down, potassium goes out, and they switch. So it's in, down, out, switch, in, down, out, switch. It's a little bit more complicated, but if you can remember all of that, then you've got the full action potential and you've got saltatory conduction there. Okay, so key points about this, a couple of key words that go with this, is if the potential doesn't go all the way to the threshold potential, nothing will happen. There won't be a new action potential. So if this sodium diffusing down here isn't enough to take this node of Ranvier, the second node of Ranvier there, to -55, then nothing will happen. It has to hit that threshold or it won't work. So if your action potential at the first gap isn't enough to bring in enough sodium for enough to diffuse down, that's where the signal stops and nothing further will happen. So it has to go, it has to take the next node of Ranvier all the way to -55, or nothing will happen. That's called the all or nothing principle. You may notice as well, a couple of minutes ago, just coming in at the far side, you'll see I've got sodium diffusing into that very start as well. So that would have been from a previous node of Ranvier, because the signal just carries on and on. What we look at in the next video, which is on receptors, is where the initial sodium came from in the first place, because it's not just nodes of Ranvier all the way along. We eventually get to the start of a neuron. So how does it all start? That's in the next video on receptors. Hopefully this one's made sense. So take a screenshot of this, because we'll now put this information into the action potential graph that we looked at in the previous video. Same graph from the previous video, so we can go quite fast on this again. This is version 2 though. Remember from the last video I said it was version 1 because we needed to add this saltatory conduction bit. So if we've still got the graph there, voltage up the side, time along the bottom, we've still got our points A, B, C, D, E, F. If we're putting the descriptions down, A, remember, was the resting potential. So that's sodium potassium pumps are actively transporting out 3 sodium ions for every 2 potassium ions that come in, making the outside more positive than the inside and setting up concentration gradients. That gives you a resting potential of -70. B, we didn't have on the last graph, but now we know that B is sodium ions from the previous node of Ranvier diffusing into the current node of Ranvier, which raised the threshold potential to -55. So if you look at the graph, I've put a little dotted line on the red, and at the side I've marked -55. So from B, point B, that increase there from -70 is caused by sodium diffusing down from the previous node of Ranvier. So from B up to where the dotted line crosses the red, that increase is caused by diffusion of sodium ions from the previous node of Ranvier. It's only after that dotted line, once we've hit -55, that we can go to C, which is the true depolarization, which is the voltage-gated sodium channels opening, allowing an influx of sodium ions by facilitated diffusion, raising the neuron in the raising the voltage in the neuron cytoplasm. That takes us to D, which is the action potential plus 40. Voltage-gated sodium channels can now close. That's important to remember for in a couple of minutes. Those channels close, the potassium voltage-gated channels can open. So at E, we get this efflux, this exiting of potassium ions by facilitated diffusion because of their concentration gradient. It goes too far though, and it takes us down to F, which is the hyperpolarization of -85. We've then, of course, got to recover by switching them back around using the sodium potassium pump, so it takes us all the way back to A. So this is now complete. So take a screenshot of this, because this is a complete full description of every point on the graph, and this is almost guaranteed how you'll be asked to explain it in the exam. So take a screenshot of this, but we're not quite done. Okay, hopefully you've got that screenshot there, because what we're going to look at now is something called the refractory period. Now, if I mark on the graph from the point marked over there to the one closer to me, and I've kind of grayed it out a little bit in the background, everything there is called the refractory period. Now, refractory is just a period of recovery time before another impulse can be carried. Now, that should be pretty obvious. So you think about a Mexican wave again. If you've gone up for a Mexican wave while you're still up, you can't do another one because you're like, oh, hang on, I haven't gone back down yet. So you've got to recover to your start position before you can respond again. That's what's happening in this refractory period. You've gone up to your action potential. The sodiums are now inside. The potassiums are still inside. You've got to get the potassiums out. But at the top of the graph there, the voltage gated sodium channels have closed. So you can't move more sodium in to further increase the voltage. And it wouldn't matter if you could, because you've already hit plus 40. So what you've got to do is take that little bit of time from the start of the blue line to the end of it to get the potassium out and then to switch them back around. And that's what takes that period of time, the potassium going out for the down and then trying to get them back to position as you go from hyperpolarization back up. Okay, now it's really important you've got this refractory period, because that ensures a nerve impulse can only travel in one direction, because it can't get excited, excite the one next to it and go, oh, that one next to me's gone, I'll come back. It's not like a Mexican wave where if the person next to you there goes up, you go up, but then the person next to you goes, oh, they went up, I'll go up again. Then it just ends up bouncing all over the place. It needs to be, it comes to you, you're concentrating on that, then you're concentrating on recovering, and you're waiting for a new one in that direction. It also ensures discrete impulses, because by forcing time for recovery, then you need it to go all the way up and all the way back down and recovered before you can have another one. It can't go up and then down a bit and always another one and end up with a graph all over, because then the level of stimulation will never be constant, would be too much to process for the brain. So it's really important you get a discrete, yes, here is an impulse, here's a stimuli. And thirdly, it limits the number of impulses that can happen to avoid overstimulation. It takes, if you look at the whole graph, it takes about 3 milliseconds, a little bit less than 3 milliseconds to have a full nerve impulse. That means you can only have an impulse every 3 milliseconds at maximum, according to the timings on this graph. Otherwise, they'd be going so fast that the brain would be receiving too much information, you'd be overstimulated, and you wouldn't be able to process the data. So the refractory period is an important thing, so take a screenshot of this one as well. Right, that's the majority of nerve signaling done there. We've got two main kind of key things still to add on to this. One is where that initial signal comes from in the first place, which is talking about receptors in the next video. And then we need to look at how the signal jumps from one neuron to another by looking at synapses. Once we've put all that together, you've got the bulk of your nervous communication. After that, we just need to add on how the nerves communicate with muscle cells, how contractions occur, and then we're going to look at specifically how the heart as a muscle can be controlled by the nervous system, or certainly by electrical signaling. As always, I asked at the start, please do give this video a like if you find it useful, recommend it to your friends, post it on wherever you want to post it. I'd love it to be used as widely as possible. Do subscribe to my channel if you haven't done so already, and do remember you can find Conscientious Biologist information on both Facebook and Instagram. Thank you.